The overall goal of this experiment is to obtain the kinetic parameters and processes of mutual chemical exchange in small organic and organometallic molecules that are difficult to measure by traditional methods.
This method can help to understand molecular dynamics of organic and organometallic molecules undergoing chemical exchange, for example, rotational barriers, conformational equilibrium, nitrogen inversion, ligand exchange, and tautomerization.
The main advantages of this technique are that there is no need to reach coalescence of the exchanging signals and that the rate constant and relaxation time are obtained in the same experiment.
We first had the idea for this method when investigating the fluxional behavior of platinum-aline complexes in solution, and we realized that variable temperature and line shape analysis techniques were not suitable.
From our experience in protein-ligand interactions by NMR, we realized that in fact similar NMR approaches could be applied to study exchange processes in organic molecules, and it turned out to be true.
Add 5 milligrams of NN-dimethylacetamide to an NMR tube appropriate for low temperatures, and dissolve in 0.6 milliliters of deuterated toluene. To set up the SSTD NMR experiments, first insert the sample in the magnet.
One of the key steps in a SSTD NMR experiment is the temperature control, so be sure to let the sample stabilize at the selected temperature for at least 20 minutes.
Once the sample is in the magnet, type EDTE in the command line. Change the temperature to the first selected temperature to carry out the experiment. Let the sample stabilize at the chosen temperature for at least 20 minutes. Perform a one-dimensional